All right, so this is a table that is in your online lab manual, and it summarizes information about a bunch of different DNA polymerases that researchers can use. Now, their names are shown here in the first column, and the names are all basically abbreviations of the genus and species of the prokaryotic organism from which they were derived. They were all identified from bacteria. Okay, we have a lot of information in this table, but what we're going to focus on initially is the exonuclease activity of these different polymerases. So DNA polymerases, some have different exonuclease activity. So what is exonuclease activity exactly? Exonuclease is the ability to remove nucleotides from the end of a strand. So exonuclease activity, you remove nucleotides from the end of a strand. All right, so to see what that means, here first we're going to use this black arrow to represent DNA synthesis. And so this top diagram is basically what we just saw in the first video, which is 5' to 3' synthesis. So that's the typical direction that all DNA polymerases synthesize. All right, now the scissors represent exonuclease activity. So here you can see the exonuclease activity is in the opposite direction. So this is 3' to 5' exonuclease activity. So we're removing nucleotides in the opposite direction that we're adding them. All right. Now this is really important because this is what gives DNA polymerases their proofreading ability. So DNA polymerase, if it puts down an incorrect nucleotide that's not complementary to the template, it has the ability to stop, chop off that nucleotide, and try and add a new one. So that 3', 5' exonuclease is really important for the accuracy or the fidelity of DNA replication. Now in our bottom drawing here, what we see is that both the arrows and the scissors are going the same direction. And so this is 5' to 3' exonuclease activity. In other words, we're removing nucleotides in the same direction that we're synthesizing the DNA. And this is important in cells for primer removal, which takes place during DNA replication. So you can imagine if there's a double-stranded region out in front of this polymerase moving along here, it has the ability to chop away those nucleotides and allow the synthesis of the new strand to continue. So that's important for removing primers. All right, so now back to the table here. Here's some of our enzymes have 5' to 3' and some have 3' to 5' exonuclease activity. So let's look at the top row, the Taq polymerase. It does not have 3' to 5' exonuclease activity. That means it can't proofread. And so, therefore, it has a fairly high error rate, which is shown here in the last column. Now we jump down to the bottom. We have a couple of polymerases here that do have 3' to 5' exonuclease activity. And you can see that relative to that first one, they have a much lower error rate. And another way to say that is that they have very high fidelity. So fidelity is just another way of saying accuracy. So 5' to 3', the 3' to 5' exonuclease activity means they're very accurate. Now, there's this one down at the bottom, which is kind of a weirdo. It's got a really high error rate, and we're just going to kind of ignore this one for now. It's a very different flavor of DNA polymerase. Jumping back up to the top here, you know, that Taq polymerase, it has a high error rate, which corresponds to low fidelity. But that also means that it's pretty fast, you know, and so sometimes there are applications in which having very fast DNA replication is all right if you're okay with a certain amount of errors.